Welcome students. In the previous class we have seen how the different how a zygote undergo changes and develops into a mature embryo, that is what are the different stages. Here in this we have seen what is growth. We have seen the characteristics of growth. That is, growth is nothing but permanent, irreversible increase in the size of the organisms, and that size increases with the increase in number of cells, and when the number of cells increases, the weight also increases. So yeah, it is a very ordered process. It is the development of plants is a very ordered process, and the first stage in the development is growth. The first stage is in the development of the organism or the plant is growth. Here growth takes place. The first step in growth is seed germination. The first step in the growth of a plant is seed germination. Development takes place by two stages. One is growth and another is differentiation. Coming to the growth in the plants. For the growth in plants, the first step is seed germination. Seed germinate during favorable conditions, and when the conditions are unfavorable, the seed undergoes the period, or that is, the seed undergoes dormancy. The seed undergoes dormancy. Here we have in the class we have seen that growth is fundamental and inconspicuous process, and the growth is indeterminate. The growth is indeterminate. Now along with this, that is, the characteristic of growth is fundamental, conspicuous, the growth is indeterminate, and another characteristic of growth is measurable. Most of the times the growth is measurable. We can measure the growth, but at cellular level, here growth can also be seen at cellular level. Here what happens at cellular level? How can we say the growth is taking place? When the increase in protoplasm takes place in the cell. If we see growth at cellular level, so when can we say the growth is taking place inside the cell? When the protoplasmic content increases. When the protoplasmic content increases, we can see that increase in the protoplasmic content of the cell, but we cannot measure it. We can see growth at cellular level. Earlier cell was only this much of size, for example. Now it is of this size. This increase in size is because of the protoplasmic content. Increase in the protoplasmic content. We can see this increase in the cell. We can see this increase in the protoplasmic content of the cell, but we cannot measure it. Why? Because we does not have such sophisticated and sensitive instruments which can measure the protoplasmic content. We can see it, but we cannot measure it. But there are different parameters. There are other parameters which shows that growth is measurable. We can measure growth with the help of a variety of parameters. With the help of variety of parameters other than this at cellular level, here we can show that growth is measurable. We can show that growth is measurable. Here growth is measured by a variety of parameters. Those parameters are nothing but increase in the weight. Increase in the weight. You just take when the fruit is on the plant itself, when it is fresh itself, you just weigh the weight of the fruit. You just weigh the weight of the fruit. Here it will be 1 gram. It will be 1 gram. When it is still on the plant, it will be fresh. It will be 1 gram. After some time, after few days or after some month, like you go and again weigh it will be 2 gram or 3 gram. It will be 2 gram or 3 gram. So here this shows that weight has been increased. That increase in weight, I said you, when the increase in number of cells takes place, then the weight of cell will also increase. So this shows when the weight is increased, When we see that there is increase in the weight of that fruit, here we can say that the growth has taken place. Okay, by checking the weight we can say growth has taken place. So, we can measure by weighing the weight of the fruit or any other part of the plant. Okay, and length, we can say when there is increase in length, by seeing the increase in length of the plant, length of organ, we can say growth has taken place. For example, length of the pollen tube, earlier it was only of this size, earlier it was only of this size. Go after few days and again measure it, go after few days and again measure it. Now it will be of this size, it will be of this size. We can clearly see this increase in the length, we can clearly see the increase in the weight. So, increase is nothing but we are measuring it. How we are measuring it? By seeing the increase in the weight, by seeing the increase in the length, by seeing the increase in the length. We can also see increase in area. We can say when the area is increased, we can say growth has taken place. We can measure how much of growth has taken place by seeing the area. In the sense, for example, leaf expansion. Leaf expansion. When we take the example of increase in area, then we can take the example of leaf expansion. Earlier the leaf was of this size only. After some days you go and check, now the size of the leaf will increase. So, by this we can measure the growth. Earlier growth was this much, now it is this much. We can measure by checking the area, increase in the area. We can measure that is the different parameters. What parameters we are taking in order to measure the growth? We are taking weight as one parameter, then we are taking length as one parameter, we are taking area as one parameter. When there is increase in the area, surface area, then that means growth has taken place. When there is increase in the length, then that means growth has taken place. So, in order to measure growth, we take different parameters. Those parameters are weight of the organ or whatever we are taking weight, leaf ka weight layer, taking the weight of the leaf or taking the weight of the stem or fruit, whatever. We can even say this increase in the volume. Volume can also be taken as one parameter for measuring the growth. For example, to measure the volume, what we'll do? Take a beaker, put some water till 50 ml. Put the water till 50 ml. Then take one apple, dip it. Now what will be where will be the level of water? The level will be showing at 80 ml. The level will be showing at 80 ml. So that means what is the volume of that apple? 30 ml. 30 ml, because already 50 was the volume of water. Now it is showing 80, that means the 30 ml which has been increased is because of the weight, that is volume of the apple. Then increase in cell number. When we count the number of cells also, we can say that growth has taken place. Here one example, one amazing example here is single mature root meristem of maize plant can give rise to 17,500 new cells, can give rise to 17,500 new cells in just 1 hour. So, when there is increase, before 1 hour the cells were very few, after 1 hour this much of increase has taken place. So, by seeing the number of cells we can say that growth has taken place, and we can see the size also. When the size has been increased, the size will be increased because of the addition of the new cells. So, in watermelon up to 30,50,000 cells will be increased, that is will be added, which will show the increase in the weight of the watermelon. So, what we are trying to say is weight can be measured, growth can be measured, growth of the plant can be measured. How can we measure the growth of the plant? By taking different parameters. What are those parameters? By checking the size, by checking the length, by checking the number of cells, by checking the area, by checking the volume, by checking the weight. These are the different parameters through which we can measure the growth. At cellular level also, growth will be seen, that is increase in the protoplasmic content, but it can only be seen at that level. We can only see, we cannot measure, because we does not have that sophisticated and sensitive instruments. So instead we can measure the growth by this following parameters. So we have seen the characteristics of weight, that is growth. Growth is fundamental, growth is conspicuous, growth is indeterminate, that is unlimited, and growth can be measured. Growth can be measured. Now we will see the different phases of growth. The different phases of growth. Growth takes place in different phases. It will not take place at once, instant, constant. It will take in different phases. Here the first phase of growth is meristematic activity. Meristematic activity. The second phase is elongation. Meristematic activity in the sense production of new cells. Production of new cells. The cells will be continuously, meristematic cells will be continuously dividing and producing many new number of cells. When cells are added, then that is growth is taking place, and after this meristematic activity, here the growth enters into second stage, that is the elongation phase, and next is the third phase, which is called as maturation phase. Maturation phase. Growth takes place in three phases. First is meristematic activity, second is elongation activity, and the third is maturation. Coming to one example we can take for this, all to show all this. Root tip. We'll take one root tip. This is the root tip, and make these markings here, not root tip. You just take this newly germinating seed. Here mark these markings. At some levels you just mark it. So here it will be near, because this is small now. The markings will be very near. Leave it for some time. Then check it again after some time. Here this A, in between this A and B, at earlier, before some time the space was very little. Now space is more. Here between B and C, very little space was there. Now here the space is more. How can we say the space has been increased? Because we have put the markings. When this has grown, so here it was shown. When we measured the distance between the markings, it showed that growth has taken place. It showed that growth has taken place. Now here, coming to this, I said you three phases. First phase is the meristematic activity. Meristematic activity, that is this activity which is done by meristematic cells. The activity which is done by meristematic cells. We all know meristematic cells are the rapidly and continuously dividing cells. Meristematic cells are the rapidly and continuously dividing cells which produce new cells. These cells are present at the shoot tip and the root tip, apex, the top region of the root and the top region of the shoot. This meristematic activity is because of meristematic cells. Meristematic cells. These meristematic cells, they are present at two regions. The first is the tip of the root and the second is the tip of the shoot. Apical meristems, that is the meristematic cells which are present at the tips of roots and shoots, those are called as apical meristems. So these apical meristems, that is meristems which are present at the tip of the root and tip of the shoot, they continuously divide and produce new number of cells. For example, here is the meristem. Example I am saying. So it will continuously divide and produce new number of cells. So when new number of cells is there, then it will show increase. So this meristematic activity, here there are dividing cells, continuously dividing cells will be present, and these cells will have, these continuously dividing meristematic cells will have good protoplasmic content. It will have good protoplasmic content, that is the content inside the cell, because the protoplasm, that cytoplasmic content of the cell is very important, because that cytoplasmic content will have all the enzymes. So, more the cytoplasmic content, more the enzymes, more will be the activity. So, they can, those meristematic tissues or the meristematic cells will have enough protoplasmic content and they have large nuclei. They have large nuclei. Large nuclei because we all know nucleus is the main organelle which coordinates the functions of the cells. Nucleus is the main organelle which coordinates the functions of the cells. Then those cells will also have thin cellulosic wall, because if the wall is thick, and it is made up of some impervious substances, then it will not be able to divide. It will not be able to divide. The walls will become very rigid. It will not be able to divide very easily, very rapidly, and in very shorter time. So that is why the cells, the first activity in the growth is the meristematic activity, that is this meristematic cells will continuously divide and produce many number of cells, and those cells will have enough protoplasmic content, they have large nuclei, and their cell walls are thin. The cell walls are thin. Now after this tip, for example, if this is the root, this is the tip, here meristematic cells will be present. Just above this, that at the proximal, that is just above this meristematic cells, we have the zone of elongation. The cells, the newly produced cells, here they move upwards and they start the process of elongation. They start the process of elongation. These, they are just producing many number of cells. Now many number of cells here, they are being pushed upwards because they will produce more cells. So when they push upwards, then only these meristematic cells will be able to produce, will be able to give space. So here the cells will be continuously, the meristematic cells which has been produced will move to this region. This region is called as zone of elongation. Here the cells just above this, at the proximal, that is just above the meristematic region, apical meristems, we have zone of elongation, and the cells which are present, meristematic zone of elongation, the cells which are present in zone of elongation, they have high vacuolation. Big vacuoles will be present in them. Big vacuoles will be present in them. The cells, the meristematic cells which has moved from meristematic region to the zone of elongation, now the cells will enlarge. The cells will enlarge or elongate, and the cells will have high vacuolation, that is many number of big vacuoles will be present. Why? Because to undergo the process of elongation, energy is needed, and for energy food is needed. These vacuoles will be storing the food material. These vacuoles, they will be storing the food material. So for the zone of elongation, the cells will have high vacuolation for food storage. Then enlarged cells. Earlier the size was small. Now here when it moves to the zone of elongation, the size of the cell will also increase, elongate, and here this increase in size, protoplasmic will be there, vacuoles will be there, and new cell wall deposition. Here the cell wall was very thin and cellulosic. Now here around the cells, here a layer, a deposition will be seen. A layer or the deposition will be seen. Deposition of lignin, suberin, pectin, many are substances. So deposition will be seen and the cell wall becomes thick. Here the meristematic cells was having thin cell wall, but here when they enter, the cells will have thick cellulosic as well as lignified thickenings will be present. The cell wall will become thickened. Just now after this they have elongated and all elongation of cell has taken place. Now these cells will move towards this upper region. Just above the zone of elongation will be zone of maturation. Just above the zone of elongation will have zone of maturation. Here these cells, when these cells move from here to here, now these cells will have maximum size. The cell will reach its maximum size. Here the cell was very small. Here the size has been increased. Now these cells will attain the maximum size. These cells will attain maximum size and the cell wall become thick. The cell wall become very thick and these cells will change into these meristematic cells. Now these meristematic cells will convert into permanent tissues. These meristematic cells will convert into permanent tissue. When they have attained the maximum size of the cell, when they have proper thickening and all is formed, now the cell has matured. Now the cell has matured, properly formed. Now after the formation, now it will convert into permanent tissue. It will convert into permanent tissue and here protoplasmic modification will also be seen. For example, if this cell has moved into this maturation, increased in size. Here thick cell wall has been formed. Thick cell wall has been formed around the cell. Now this will change into permanent tissue. For example, if it forms trachea. Trachea is the cell which is present in xylem. Xylem is a tissue which conducts water. So the trachea is a cell in xylem which conducts water. Here it is a dead cell. Trachea is a dead cell. For example, if this cell is changing into a trachea, if this cell is forming a trachea, then what will happen? Protoplasmic modification will take place because in trachea protoplasmic content should be less. Trachea is nothing but a dead cell. Protoplasmic content should be very low. So what will happen? This cell loses its protoplasmic content. The cell loses its protoplasmic content and become dead cell. For example, if this cell changed into parenchyma, instead of trachea, it changed into parenchyma. Parenchyma is a living tissue. Parenchyma is a living permanent tissue. It needs protoplasmic content. So what happens to the protoplasmic content? It will be as it is because the cell needs it. If it is changing into trachea, the cell does not need any protoplasmic content. So the cell loses the protoplasmic content. So this happenings which takes place in the protoplasmic content, that is called as protoplasmic modification. So when the cells convert into permanent tissue, protoplasmic modification will be done and the cell will be differentiated. The cell will be differentiated, that is assigned a proper function. It will be assigned a proper function. In the next class, we will see what happens, what is differentiation. In this class, we have seen what is growth, because we said development takes place because of two phases. Development takes place in two phases. One is growth, another is differentiation. We have seen growth. We have seen growth, what is growth and what are the different characteristics of growth, and we have seen the different phases of growth. Now the cells have been totally grown and matured and changed into permanent tissues. So what happens after the cells mature? After cells mature, they undergo a process that is called as differentiation. That is the second step of development. First step of development was growth. Second step of development in plant is differentiation. In the next class, we will see differentiation.